Assalamu alaikum everyone. Hope you all are doing well. This is me, Aisha Zahid, your science teacher, and I welcome you all in the new session, in the new term. Hope that the midterm exams have gone well, and we are starting our final term session with the best wishes and all the positivity. And today we are going to discuss the ongoing chapter of class 5, which is flowers and seed. So let's get started. This is your book for grade 5, which is published by Cantab. And the chapter that we are going to discuss, the chapter that is ongoing in our class, is flowers and seeds. So, up till now, we have discussed the basic concepts about the structure of flower, the structure of seeds. We have discussed about reproduction. We have discussed about the type of reproduction and so on. So, let's have a little overview of all the things and then we'll move towards the new topic. First of all, we have discussed about the structure of a flower. A flower is basically a very a distinguished feature of a plant. It is a very beautiful part of a plant, and flowers are the reproductive organs of a plant because they develop into fruits and into seeds lately, right? So flowers are the part of a plants that are converted into fruits and seeds. That's why they are known as reproductive organs. Then we have discussed the four main parts of the flower, that is sepal, that is petal, that is stamen, and that is carpel. You can see in the figure 3.1 we have differentiated four different parts of flower. First of all, it's sepal, which is the green part. The green leafy structure is known as sepal. Then we have petals. Petals are the colorful parts of the plants. Then we have stamen. Stamen is also known as like the pollen grains, the anther and the stigma. The red seed type structure is the stamen, and then we have carpel, the whole part, the whole flower is carpel, right? The internal structure of a flower is carpel, and which includes ovary and style and stigma. We will discuss it in we will describe it lately. First of all, we will discuss that what is sepal. So sepal is basically a part of a flower which is covered by leaves. As we have discussed that it is a green leafy structure, and the main function of sepal is to protect the flower when the flower is in a bud stage. You might have observed that we use the word kali in Urdu for the bud stage, right? So when the flower is in the bud stage, this sepal is responsible for the protection of a flower. Then we have petals. In Urdu we used to say, what would be the word for uh, like the flowers, the petals are the colorful parts of the plants flower, right? जिसको फूल की पत्तियाँ कहते हैं, ठीक है? उर्दू में पत्तियाँ वाला वर्ड इज़ यूज फॉर द पेटल्स. So the colorful part, the most prominent part of a flower is the petals, and the main function of the petal is to attract the pollinators. We will discuss the phenomena of pollinator lately. So the main function of petal is to attract the pollinators, right? Then we have stamens. So stamen is like a third whole complete part of a flower, which includes a long tube-like structure known as filament, and it's the male part of a flower, and it has pollen grains present on the top of the filament. In this structure, the red tube-like structure is stamen, and the bead type red structure is known as pollen grain, which is attached on the top of the filament. Then we have carpel. Carpel is basically a female. We can say that the carpel is a female reproductive. Carpel involves a female reproductive part, which is ovules, and that also known as eggs, and they are responsible in the sexual reproduction. Then we have discussed in the class about the pollination and types of pollination. So uh, what basically happens is flowers used to reproduce themselves by different processes, and there is a process known as pollination in which the pollen grains of one flower are transferred into the pollen grains of an other flower. Like basically what happens is the male part of a flower transfers to the female part of a flower. The male part is known as anther and the female part is known as stigma. So basically anther is transferred to the stigma and then they fuse together, and then new flower, the new seeds are formed. Okay, so there are different types of pollination. First of all, we have self pollination, then we have cross pollination, then we have like or different pollinations, right? Then we have discussed how the pollen grains are transferred. So there are different modes of transferring pollen grains, whether it's through air, whether it's through birds, whether it's through water, whether it's through wind, and so many other things, right? Okay. Then we have discussed about the reproduction and its types. So basically reproduction is a process through which the new young flowers are generated. Okay, so plants are also reproduced through living organisms, and there are two types of reproduction. One is asexual reproduction and the other one is sexual reproduction. Asexual reproduction is a mode of reproduction in which there is no need of a seed. There is no involvement of a seed. Okay, what basically happens is the parts of a plant, the parts of a like the whole plant, leaves, stems and roots are converted into a new plant. It's known as asexual reproduction. The word a represents no. In biology, in science, the word a means no, not, and sexual means sexual reproduction, like fusion. So asexual means there is no fusion of male and female gametes, right? Male and female parts of the plant, which is anther and stigma. And in sexual reproduction of plant, what basically happens is uh, there is a fusion between the sperms and the eggs. Okay, after the fusion, the new seed used to grow. This is sexual reproduction. Then today's topic is about the structure of monocot and dicot seeds. Now we have discussed about reproduction. ठीक है, so the basic difference between sexual and asexual reproduction of plant is कि sexual reproduction involves seed and asexual reproduction do not involve seed, right? So seed is very much important for the reproduction, and seed is a very much important part of a plant, right? So we have two types of seed. The one is monocot and the second is dicot. Okay, first of all I wanted to make it clear that um, mono means one and di means two. Okay, so the basic difference of monocot and dicot plant is कि monocot plants have one cotyledon and dicot plants have two cotyledons. Now you guys will be confused at what are cotyledons. So let me show you a picture of cotyledons. This greeny leafy structure is known as cotyledons, right? These two. Now the seed that have only one cotyledon is known as monocotyle, monocots, and the seeds that have two cotyledons is known as dicot. This is the picture of a dicot seed in which two cotyledons are present. Okay, so this is a dicot plant. So okay, seed is the product of fertilization. When male and female gametes are fused, are fertilized, they convert into seed. Okay, we have two types of seed. We have on the basis of cotyledons. Now if someone will ask a question to you that what is a basic difference, why the two seeds are differentiated, the answer would be the two seeds are differentiated on the basis of the cotyledons. We have discussed the structure of cotyledons कि क्या होते हैं cotyledons. You would say कि in monocot plant there is only one cotyledon, and in dicot plant there are two cotyledons, right? First of all, we will discuss the structure of monocot seed. So, monocot seed is basically composed of seed coat, endosperm, and embryo. Seed coat is also known as testa, and seed coat is basically is a outer covering of a seed, बिल्कुल बाहर की साइड पे, which is helps to protect the embryo. ठीक है, basically seed coat protects the embryo from any sort of injury and any sort of damage. That's why the thick seed coat is present around the seed, which is not easily removed. It is very thick and it is very hard. Then we have endosperm. Endosperm is a yellow colored tissue, ठीक है, in monocot plant that are responsible for the supply of food. ठीक है, monocot plants have endosperm, which is a yellow colored tissue, and they are responsible for the supply of food. Then they have embryo. Embryo is a part of seed where basically the new seeds and the new plants are generated. In other words, embryo is a baby plant. Baby plant, like inside a seed there is an embryo which develops into a new plant. You might have observed that the seed breaks and a new plant germinate from the seed, right? So the baby plant that germinates from the seed is the embryo. Embryo involves plumule, radicle, and cotyledons. We have discussed the cotyledon earlier. Plumule is basically plumule is a part of embryo that grows into the stem, and radicle is a part of the embryo that grows into roots. Okay, and uh, in the whole plant, the first thing that is visible is a radicle. Like first of all, there is a development of roots, that is the radicle. Then we have the structure of dicot seed. Dicot seed involves seed coat, cotyledons, and embryo. Seed coat again is a outer covering, outer part of seed, which protects the embryo from the damage and the injury, from any harsh condition, from infections, and so on. Cotyledons. Dicot plants have two cotyledons, ठीक है, and dicot plant do not have endosperm. Monocot plant have endosperm. Dicot plants do not have endosperm. It means that the food supply of dicot plant is through cotyledons, right? Monocot plant have a food supply through endosperm, and dicot plant has a food supply through cotyledons. Now we have embryo. Embryo is again a baby plant inside a seed, and the embryo of dicot seed involves radicle and plumule. Then there is a basic difference between monocot seed and dicot seed. Monocot seed involves one cotyledon, dicot seeds involve two cotyledons. Monocot seeds are small and are very thin and do not store food. Dicot seeds are like fleshy, like thick, and they used to store food through through two cotyledons, right? Monocot seeds have endosperms, dicot seeds have do not have endosperms, right? Then monocot seeds have radicals, and dicot plants uh, do not have radicals, right? Then there is a process known as germination. Germination is a process by which a new plant germinates, a new plant grow inside the seed. The moment the seed is broken, like the seed is breaking and then baby plant is germinating, this process is known as germination. So we have two types of germination: apigeal germination and hypogeal germination. Apigeal germination is a type of germination which occurs above the ground, which is visible, right? But hypogeal germination is a type of germination which is present inside the ground, right? So hypo means below and geal means earth. This is a type of germination which is occurring inside, like below the ground. The common examples of hypogeal germinations is maize, pea, rice, gram, groundnuts, and so on. So what basically happens is, in the hypogeal germination, the seeds remain inside, like below the soil. The seeds are usually below the soil. Cotyledons do not play any role in monocot seeds. Obviously they do not store food. There is a greater elongation of the epicotyle. Epicotyle is also a part of a plant. Okay, you can see there is a growth of the growth like structure, it is known as epicotyle. Then we have a terminal, which is an end part of epicotyle that is little bit curved. Okay, so then we have basically the conditions that are necessary for germination, like what are the factors that will increase germination and what are the factors that will affect germination. So the germination of seed basically depends upon water, oxygen, sunlight and temperature. Water is very much important to trigger the process of germination. Like more there would be water, more the seed will be softened and it will easily rupture and will easily broken down, and the new plants will easily grow. Oxygen and air. Germination of seeds needs a continuous supply of oxygen, as the germination seeds get energy from the stored food, and this process is known as respiration. And respiration occurs in the presence of oxygen. Then we have sunlight. Sunlight is very much necessary for the germination of seeds, and low sunlight will obviously affect the process of germination. Then we have temperature. There is a desired temperature for the germination of seed, which lies between 15 degrees Celsius to 30 degrees Celsius. The fluctuation in the temperature will obviously affect the germination. Now there is a difference between dicot and monocot. You can easily observe that, look, this green structure has two cotyledons, that's why it's dicot plants. And this green structure has only one cotyledon, that's why it's monocot plant, right? Then we have discussed it's a cotyledon. Okay, the two green parts, that's why this is a structure of a dicot plant. I hope this chapter was quite interesting and quite understandable to you guys. If you guys are having any question and any query regarding any term, regarding any concept, regarding any um, process, regarding any um, part of a plant, you are always welcome to come and ask and clarify all the questions and all the concepts, right? So up till the new topic, up till the new chapter, best of luck and enjoy your winters, and keep studying, keep working hard, and best of luck. Thank you so much for taking your time, and thank you so much for listening the lecture. Take care. Allah Hafiz.